just do that. All right, so this is chapter 9, and chapter 9's title is, it says Introduction to Microbial Genetics. All right, and I have to tell you that this chapter and the immunology chapters are my favorite chapters because this is what my basic training was a long time ago when I was a scientist, right? I did molecular biology and I did immunology as a scientist. So this stuff is very near and dear to my heart, and you probably have to learn a little bit more detail than if you had someone else, but we do a little bit less detail somewhere else. So I'm going to share my screen with you now, and hopefully, there it is. So this is these are the PowerPoints from chapter 9. And this one I like to include because I like to remind everybody what we're talking about when we say genome, right? So a genome means it is all of the DNA that the organism contains. So a cell's genome does include, for example, here's a eukaryotic cell with some plasmids, and here's a bacterial cell with a plasmid. All of this is the genome, even the plasmids, because they do have the ability to produce proteins, which can then influence the activities of the organism. I would again remind you that eukaryotic chromosomes are linear, and they're arranged in linear segments, pairs usually. Most cells are 2 ends, they have 2 copies of each chromosome. This is a mitotic chromosome. These are mitotic chromosomes here that have already been duplicated, and they are, of course, inside the nucleus. Notice they're showing you where there is DNA inside a eukaryotic cell. Fungus and some protozoa can have plasmids, and remember that inside the mitochondria and inside the chloroplast, there also is DNA, and remember, it's not as this pictures it, it is circular, not linear like they've drawn it. Inside the prokaryote, we have this single chromosome. Notice it's twisted, double-stranded DNA, double helix, in addition to some plasmids, potentially maybe. And then in viruses, they have multiple variations of genomes. Some of them have DNA, and as this one is depicted here, it has double-stranded DNA. Some viruses actually have single-stranded DNA, and other viruses have RNA. Some of them have double-stranded RNA, some of them have multiple strands of RNA that's used as genome. So I want you to remember that predominantly genome, when we say that, we mean DNA, but if we're talking about viruses, we're talking about DNA and different kinds of RNA, and there's much more variety in viruses. And this packet just goes through the list. This picture is not in your most current edition of the book, but it is my favorite picture of all time in microbiology, so I have to include it. I used to have a t-shirt with this on. Okay, there, so that's how nerdy I am, right? It was from a company that was giving away free samples on how to get the DNA out of a bacteria. So they were using it as their as their poster. Anyway, all of this is the DNA, the chromosome from inside a single E. coli, right? And so when we draw the chromosome to look like this, right? Remember, that's like a schematic, an artist's rendition. In real life, it's a lot messier, right? And it takes up most of the space. If you remember back to chapter 4, we had those pictures that showed us how much actually of the cytoplasm the DNA actually took up, which is most of it. All right, so now we should move on to talking about the DNA itself and the structure of the DNA. This again is something you should have learned in Bio 1, I would hope, but as always, maybe you didn't. So I'm gonna make it as large as I can. And there is a nicer picture in your textbook on page 233. It is figure 9.4. I don't think that's actually changed. I think this is figure 9.4 as well. So a couple of things I want you to look at. So I wanna look at this part of the DNA first, okay? So whenever we say the word DNA, remember that stands for deoxyribonucleic acid. And that's because the sugar is deoxyribose, which means it's missing an oxygen compared to ribose. So deoxygenated ribose, deoxyribose. Nucleic acid, because these purines and pyrimidines, right? We would refer to those as nitrogenous bases. And it's called an acid because with the phosphates on it, overall DNA itself is acidic. So that's why it's called nucleic acid, because when scientists in the 1800s first purified the material out of the nucleus, that one of the characteristics they noted was that it was acidic, okay? Purines, A and G, adenine and guanine, because purines have 2 nitrogen-containing rings. Pyrimidines, cytosine and thiamine, they have 1 nitrogen-containing ring. Remember, purine doesn't pair with purine and pyrimidine doesn't pair with pyrimidine, they pair with their opposites. So we have T, thiamine, which is a pyrimidine pairing with adenine, right? So A pairs with T and G with C. Notice the pairing between them is by hydrogen bonds. And then the outer shape to the DNA, what we makes up, like we would say, the sides of the ladder, that is phosphates bound to sugars. So that's the phosphate of one nucleotide bound to the deoxyribose of the next. So that we call that the sugar phosphate backbone, and then these would be the base pairs. Now, very important, we need to know a little bit about what we call the 5' end and the 3' end of a strand of DNA. And this this picture does not give you as great of a picture, and that's actually sort of incorrect. You can just block out that phosphate. I don't know if I can actually do that while I'm here. Whoops, sorry. Yeah, I think I need to put in a text box or something and put like a mark here. I need to put like an X over this because that hydroxide does not belong there. It won't let me. All right, never mind. Oh, I don't want to draw it on the board, but I will write out for you, especially in the notes, how to distinguish the 5' end from the 3' end of a molecule of DNA. And the 5' end of a molecule of DNA or a chain of DNA has a phosphate attached to the 5th carbon and the 3rd carbon, which you can see here, of the deoxyribose. The 3' end has a phosphate attached to the 5th carbon, but a hydroxyl group attached to the 3rd. Okay, and then the complementary strand, right? So we say they're anti-parallel because the complementary strand, 5' here, 3' there, and at the bottom, this should run 5' to 3', which there should be a hydroxyl group here, and this 5' end is correct with no hydroxyl. So I'll post a better picture of that. I'll post the one from your textbook that shows that a little bit more clearly. My apologies for using this older slide that's incorrect. But the arrows are to indicate that the left-hand strand in this case is running 5' to 3', and the complementary strand is anti-parallel, and it is running 5' this way to 3', so that the ends are not the same. Now, this is how you see DNA most of the time, right? So this is showing you the double helix. So it's double-stranded, DS for double-stranded, double helix because it forms this shape. And the reason it forms this shape is because the anti-parallel nature of the bases and the ends. And then this is the 3D model of what it actually looks like. All right, so I'm gonna put this back to normal size here and give you a second. I think I've typed it out correctly here. The 5' end of a strand has a phosphate in the 5th carbon and a phosphate on the 3rd. 3' end has a phosphate in the 5th and a hydroxyl group on the 3rd carbon. And I would just remind you that the carbons are counted clockwise after the oxygen. That's why they aren't in exactly the same places on either side of this chain, because in reality, the 3' to 5' strand, the sugar is actually flipped, as those of you who've already had Chem 2 know the answer to that. Okay, so is there anything else specifically about the structure that I wanted to tell you? No, I don't think so. I think we're good there. This table that lists the enzymes, helicase, primase, DNA polymerase 3, DNA polymerase 1, ligase, and gyrase. I believe in your version of the book, the gyrase will be listed as a topoisomerase. Okay, so there's a whole family of these enzymes. This is a little bit simpler version, and I prefer it a little bit simpler myself. So what we need to talk about next is how the DNA is replicated. And I'm going to show you this picture real briefly, and then I'm going to draw something very different on my little chalkboard, if I can, about replication. So in bacteria, because the chromosome is a circle, actually what happens is 2 sets of the replication machinery that we're going to go through actually jump on at one spot, and that spot is special, and it's known as the origin of replication, and each bacterial chromosome only has 1 of them. But see how there's 2 polymerase 3 complexes here? Because there are 2 sets of replication enzymes are going to begin replicating in opposite directions at the same time. And so if you were watching replication in a bacteria, which I don't think I included that particular slide, you would see it moving in 2 directions at the same time. And you'd get this loop that would start like this, and then it would fold out and lay way out here. This is a blow up of just one of these replication forks. This is what I'm going to focus on when I draw it on the chalkboard for you. Okay, we're going to focus on one of these replication forks, and I'm going to draw it in a linear way because I find looking at a circle doing this, I can't do it. So I'm going to put this up as a drawing on the board while we talk, and I'm going to use the names of those enzymes, but I will not have space to write out all of their functions for you. They are listed there in that table in your book. In your book, it's a purple table, and it lists the names of the enzymes and their functions. And yes, you have to know that for the test, not for the test tomorrow, but for the next test that includes chapter 9. That one will just be 9 and 6. It'll just be a little bit shorter. All right, so I'm gonna move you guys back to over here. And pardon my mess while I erase the board, get us ready for the next thing. There's a couple areas that seem to be very need to be wiped off here. It's like I need to go back to Lowe's and get some more chalkboard paint. Some scratches on my board. All right. So in bacteria, this replication process is going to be happening about every 20 minutes. In eukaryotic cells, it's not going to be as frequent. It's only going to happen during S phase of mitosis, so right before mitosis actually begins. So I'm going to draw, waiting for my board to dry here. I'm going to draw 3 strands of DNA that are being replicated, and they're going to be in different states of the replication process. Where I'm getting a lot of reflection from outside. It's such a sunny day. There, that might be a little better. Okay. So I'm going to draw one strand here, and this piece of DNA is just going to have a tiny fraction on the end where replication has started. And then this piece, sorry, I'm not doing a bang up job of this. This piece is going to be about halfway finished from what I've drawn. And then this bottom one is going to have very little left to replicate. Now on this first one, I'm going to go to the trouble of putting these lines to remind you that those are the base pairs, right? So this might be an A paired with a T or a G paired with a C. I find it, if you can't remember that, I find it easy to come up with some sort of a mnemonic device. I'll tell you mine. You can use one that's more local if you want. I can tell you a local one that some people use, but other people don't want to use. So when I was in college, I used to have to drive through Columbus, Ohio to get back home to my house in Northern Ohio. And we'd go through a place called Grove City. So I always remembered A pairs with T, G with C at Grove City, right? We live close to Gloucester City or Gloucester County, so you can do it that way too. You can say at Gloucester County or at Gloucester City, and that will help you remember that A pairs with T and G pairs with C. So this is my 5' end. My complementary anti-parallel end has to be 3', and I'm hopeful that if I follow this 5' end out here, yes, it ends in a 3' end. And whether that's true or not for my other drawings, because I didn't make them as carefully, I'm still going to label them that way. So here's where we're going to start. So we're going to start up here at the top. I know there's a big whitish spot here. I'll try to move the computer around so that it's not showing when I need to show you what I need to show you in this place. Okay. So let's start at the very beginning. So when the DNA is first opened for replication, this spot right here, this spot we call the replication fork because it looks like a fork in the road. And the bases would be already separated from each other. So the hydrogen bonds would be broken. And the enzyme that does that is DNA helicase. So I'm just going to draw it actually in my white chalk here. And I'm going to put helicase right here. And helicase's job, ASE, it's an enzyme, its job is to specifically break the hydrogen bonds, which is wonderful. But when you do that, when you break the hydrogen bonds, think about like a rope or a piece of embroidery floss. If you pull apart some of the threads to flatten them like this, what happens is you introduce coils and twists further upstream, we say, and that's referred to as supercoiling. And the supercoiling can actually break the DNA into pieces. That would be bad. So we need another enzyme or group of enzymes to relieve that supercoiling. So I'm going to draw them up here, and I'm just going to put one of them on here as a square, and it's called gyrase. Now your textbook, as I said, goes to the trouble of labeling them as topoisomerases, which is important because, like I said, there's a group of them, a family of them. Gyrase is fine for me. Okay. Come to genetics, learn more about it. You know, we can give more of the names. So we've got helicase to break the hydrogen bonds, gyrase to actually relieve the supercoiling. And somebody's chatting with me here in the chat box. I'm going to pause for a second. Yeah, I know it's not great. Oh, Gloucester County are together. Oh, that works too. That's a good one. All right, so helicase gyrase. Now we can actually get the enzymes for replication going. And the first one that we need is something called primase. And primase's job is to sit down and produce a short primer. It needs to be complementary. So if this first base was A, it's going to lay down a T. If this is a G, it's going to lay down a C. It needs to be complementary and anti-parallel. And so the pink that I've just labeled here, that's the primer, short piece of RNA. It's usually between 10 and 22, 25 nucleic acids long or nucleotides long. And I'm going to put primase then, the enzyme, right here. It's a skinny rectangle. Primase, its job is to produce this primer, and it's RNA, which is going to be a problem because we can't keep RNA in the DNA. But we'll focus on this one first. Primase provides a free 3' hydroxyl for DNA polymerase 3, which I'm going to abbreviate DNA POL, Pol 3, like that. So DNA polymerase 3, and this is the main workhorse enzyme of replication. This enzyme adds new bases super fast. So I'm going to draw it right here next to primase, even though by the time it gets here, the primase is gone. And I'm going to draw it as a circle, and I should make it kind of long and skinny. And it is going to begin sequencing DNA 5' to 3' from this place right here. This is DNA polymerase 3. Now, on my second drawing down here, I would have the DNA that was already made, and I would put polymerase 3 out here, because this is a moving target, right? We still have helicase, we still have gyrase out here upstream, unwinding and unzipping, relieving supercoiling, and the replication fork is moving. Same on my bottom diagram. Helicase is still here, and gyrase is still 5' of that, or upstream of that. Here. There. Hold on. Now, this primer, we need to get rid of it. It can't stay, that little piece of pink, right? The enzyme that gets rid of that and replaces it with DNA, that enzyme's name is DNA polymerase 1. And so DNA polymerase 1, oh, I don't know what shape to draw that. I can really only draw a triangle, circle, square. So maybe we'll make that one a triangle. DNA polymerase 1, its job is to remove the primer and fill it in with DNA proper so that there's no gaps. Polymerase 3 is a very good proofreader. So it goes along and it can also check that it's not made any mistakes. If it makes a mistake, it leaves a gap, and polymerase 1 usually fills in the gap. DNA polymerase 3 can work in bacteria between 800 and 1200 nucleotides a second, and in human cells and yeast cells in the neighborhood of 600 to 800 nucleotides per second. So it's going super fast, super fast. Now, as I drew this picture, what I realized is I forgot something, which I apologize, but I forgot in here in brown, I'm going to put a couple of these brown dots here at the replication fork. These are proteins. I don't think they're in your list. That's why I forgot about them. SSBP, super important. SSBP, B as in boy, P as in Paul. Single-stranded binding proteins. They're usually in pairs, like I drew them here.
and they hold the replication fork open. So I'm going to draw them on all of my replication forks because they should be there. And there might be a single pair, two, there might be as many as 4 pairs depending on the organism. And that changes, again, depending on the organism. So let's the whites back a little bit. All right, so helicase breaks the hydrogen bonds. Gyrase relieves the supercoiling. Single-stranded binding proteins hold the replication fork open so the enzymes can get in there. Primase hops on and makes a short RNA primer, which I'll label this RNA primer. Then primase is done on the leading strand for us in terms of microbiology. DNA polymerase 3 hops on the end of that primer and starts synthesizing new DNA 5' to 3'. That primer is removed by polymerase 1, DNA polymerase 1, and filled in with DNA 5' to 3'. And now we have this continuous strand here. So this strand is actually, trying to make that spot go away. It's actually just worse. Right. This strand here, we call that one the leading strand because it's made continuously, complementary and continuously. And I'm going to put DNA polymerase 3 out here at the replication fork again. Lovely. The difficulty comes, we have to replicate the other strands. And all of these enzymes only work 5' to 3'. So we cannot sit down here with primase and make another primer toward the replication fork because we have a 5' end here. So this is where the lagging strand, or how the lagging strand is synthesized. We have a primer. Primase makes a primer here at the replication fork going outward, away from the replication fork. So I'm going to put the enzyme here again, square. And this is the primer, but it's at the replication fork toward the end. Like that. Right? So on my second drawing, we would still have the primer would be here, right? But DNA polymerase would be jumping on here and synthesizing, and I'll make it a little bit longer, 5' to 3'. But the primer is still sitting there, like that. This piece, I can't make it so that you can see that. Let me try a book. This piece, there we go, a little better. This piece is called the Okazaki fragment. Step away. Sorry. Is that important? This piece here. Okazaki fragment. They are usually about 200 base pairs long. I'm going to call this one fragment A because this is going to repeat. We're going to have primase is going to make another primer here at the replication fork. DNA polymerase 3 is going to hop on and fill that gap till it gets to there, and then we're going to need polymerase 1 to put those, to get rid of this old primer, right? So that we just have a long piece. I'm going to make it down here in purple. We have 2 long pieces now. This first one, fragment A, and this second one, fragment B, that are all DNA, right? Because remember, polymerase 1's job is to get rid of the primer. So I kind of smudged that there, but I'll do that. And the last enzyme we need is one to join the fragments. I'm going to make that a bluish diamond. And that enzyme's name is DNA ligase. This segmental synthesis of the bottom strand here, of the lagging strand, is still complementary, it's still anti-parallel, but we do it in these pieces of Okazaki fragments that then get joined together by DNA ligase. Again, because polymerase 3 is so fast, it can do all of this in, if you're a bacteria, it can do all this in 20 minutes. It can copy the whole bacterial genome. Now, there are tons of videos for that out there that you can watch from wherever you like. I would recommend Mr. Anderson. It's called Bozeman Science. He does a great job of replication. He uses all the appropriate words for replication. I will also post my replication lecture from Bio 1 from the summer, which might help because I go a lot slower. And if you didn't get a lot of this in Bio 1, it's going to be important that you sort of catch up.